So I'm really happy to be here. And if I can get this technology to work for me, I'm going to tell you what it is that I think about CMT and how it is that I think that this group that we've assembled under STAR can help everybody in this room. So this is one of two most important slides that I'm going to talk about today. So this is a person that I didn't see personally, but was involved in figuring out what kind of CMT he had. And he's taken off his clothes, so unlike Pat's sock-covered leg, you can see that this man's been affected by a neuropathy that's robbed him of the strength in his lower limbs. And if we were to examine him, the sensation that he has in his legs. So he has CMT because he doesn't have sensory and motor function, normal sensory and motor function in his lower limbs. And basically, people have known about patients like this for a long time. Charcot-Marie-Tooth were around more than 100 years ago, finding out that there were families in which this characteristic thing happened. And they didn't even know about genetics, they just sort of knew it was the familial thing. Susan, you're making a sign. What are you? That is up. All right, I'm going to try not to make this go any harder. So the patients that have been known for a long time have seen doctors like me for a long time, and basically we did what we could when we could, but to echo on a couple of Pat's comments, it's only been since we could find the genes that cause these diseases that we would really have any chance of making them better. And as that process went forward in the 90s, people like me got involved because we thought this was going to be the coolest thing ever, to be involved in figuring out what's caused somebody's problem, and then hopefully have that be the foundation of fixing it. And so that's really where I think the motivation for STAR comes from. That's why Pat can corral me into Saturday afternoons and all-day meetings anytime he wants to, because basically me, like many other people, we're all in. And I would say this is the second most important slide. What I want to tell you is that in order to do this, people who are here with me on Saturday, everybody needs to be involved. So the people that have CMT and their families need to be involved because they're the people that we need to see to find the causes of the CMT. That's for the first thing. We got to find the causes. And we haven't found all the causes of CMT, and so there's a very active effort underway that I participate in to find causes of CMT. So if anybody out there hasn't found the cause of CMT working with their neurologist, then I'm just going to say as a side note here, you need to go to a center of excellence and get seen by somebody who can help you find the cause of your CMT. Simple as that. Because no one's going to be able to fix your CMT unless we know what kind it is. And so we found all the common causes, and those are the ones we're focused in on trying to find treatments for. But you know, I had an opportunity to find a patient who had a CMT-like thing, only he had deafness and hearing loss to go along with that. And it turns out he had a vitamin deficiency. And so his hereditary neuropathy was caused by vitamin deficiency, and we gave him vitamins, but at age 50, I couldn't make it better. But they found people just like him in this way, and if you treat them as infants, you make it better. So everybody needs to be aboard finding the cause of their CMT, in my opinion. And if you know the cause of the disease, then that gives you a chance to get the scientists of the world involved, because there are a lot of very bright minds. John Severin's here with us, who aren't physicians, they're not the ones seeing the patients, but you tell the scientists that we found that gene X causes disease Y, and you've unleashed the power of modern science to get interested in, well, how could that be? How can I understand that? I'm going to think about that. I'm going to read some papers. I'm going to do an experiment. And if that happens, then we're talking about figuring out why the disease happens, and that is the key pivotal changing point, where if you can figure out what you think is wrong with the disease, and you can make a model of that, you can make a cell model that Pat was explaining, you can make an animal model by altering the disease state in an animal like a mouse, then that gives you a chance to find a treatment. So we have, for instance, and I'll go through some examples, we have cells that have CMT, if you will, and we have mice and rats that have CMT for sure. And those animals then give us an opportunity not only to investigate more deeply the causes of the CMT, but to actually, if we think we have a treatment, treat the animal. Like, well, I'm not going to treat a patient until I've shown that something works in an animal. And that's proven in many, many diseases to be the key pivotal moment in science. You got a disease, you've got it in an animal model, you think you've got a treatment, you treat the animal, you make the animal better, Well, that's a very powerful, persuasive argument to make. And I'm going to show you an example where we've cured CMT in a mouse, and if we could only follow those footsteps, we could certainly cure CMT in a person. And then if that's all going to happen, then we're back to you again, because what we'll need from everybody here with CMT is you got to participate in a clinical trial, because we're not going to know that a treatment that we think works in a mouse is going to work in you. And to do that, we need patients who have that particular kind of CMT to come see the docs, you know, get registered, get studied on a regular basis, take the medication, which is going to have some theoretical risk to it, I presume. And in that effort, we'll finally understand whether we have a treatment that really works. And if we could complete that circle for one of the CMTs we're going to talk about today, I'm going to die a happy man. I'm going to be thinking that the professional part of my life has been fulfilled, because when I started this 30 years ago, I had no idea that things would progress this fast. Not for 1 minute did I think we'd find all the genes that cause human disease, we'd be able to investigate how they cause human disease, that we'd be able to make animal models with amazing technology, and then to finally give patients the opportunity. And it all started with the patients, and if this circle is complete, it all ends with the patients. And along the way, those bright young faces were some students, undergrad students at Penn that worked in my lab, one of whom has CMT, and I'm not telling you which one. And you got to train the next generation. So I need the next generation of scientists, I need the next generations of physicians, I need the next generation of people who give a damn. So that's really what I want to talk about today. I think that we have the opportunity, everybody in this room has an opportunity to contribute to this story. So this is my throwaway slide. This is to show you that the nervous system, the nerves, which are those blue things, I don't have a pointer, so you're just going to take my word on it, that blue set of things, those are the nerves, and the diseases of the nerves are what cause neuropathy. And CMT is basically, there's an inherited reason for things to go wrong causing neuropathy. For the kinds of CMT that cause slow conductions, so if you've been told you have CMT1, that means your conductions are slowed. That's a problem in the cells making that blue myelin sheath, and we're going to show you a picture of that in a minute. And if it's a problem that's CMT type 2, then that cell body sticking up way at the top of the slide in the spinal cord, that's where the problem lies. It's that cell's not doing its job, and so you get an axonal CMT out of that. So this thought is very robust, because if we're going to fix the problem, in one case we want to fix the Schwann cell because the Schwann cells are having the problem, and in the other case we got to fix the neuron. So I think John Sveren's going to show even a cooler slide than this, but this is my analogy. So when you look at a nerve, you're basically looking at a cable where there are many different kinds of wires. And so the wires that make your sensation work, we'll just say those are the ones that are yellow and red, and the ones that are white are the ones that make your muscle work. When you have CMT, you're basically saying both kinds of wires are affected, the sensory and the motor. So you have both numbness and weakness. But there are, and maybe somebody in this room has one of them, there are forms of CMT that just affect the motor side. And so your sensation's perfect, but you don't have the strength in your distal muscles. And there are even rare forms of CMT that affect just the sensory. So your strength is fine, but you can't feel your hands and your feet. And you might say that those are rare forms, but if it's a form that I had, it wouldn't be rare to me. It'd be the form I'd be all about. So even the rare forms are things that we want to pay attention to. So this is a real picture of a real nerve from a real patient. They had a nerve biopsy. I don't know why, because it's a normal nerve. But basically what I'm showing you here is that when we get to look at some of the slides that John Scherer will show, that there are nerve fibers. Those are the white profiles of the Oreo cookie, and the chocolate wrapping around them is the myelin. That's what the Schwann cell is supposed to make. So we can picture that the disease is either going to mostly affect those white axons or the chocolate outer wrappings. If we look real high power with a cool machine called an electron microscope, you can actually see their structure there. So the white axons have these little dots in them. I think you can see that. I wish I brought my own pointer today, but I'd be showing you that. And the chocolate outer wrapping is the myelin. And so there are diseases, and we're going to talk about CMT2E, that are affecting the neurofilaments, those little dots inside those axons, and it's a problem with the neurofilaments that causes CMT2E. And the problem with CMT2A, which John's going to talk about, are the structures in the cells. I'm just going to, you know what? I'm going to come over here.
Ready, go.
Those are the neurofilaments. That's the problem with CMT2E. You got a neurofilament problem. These are mitochondria. These are the little energy producers inside the axons. And if you have CMT2A, the problem is with the mitochondria. They're not simply making enough energy for the axon. And if you have CMT1 or the CMT4, then the problem's in the myelin sheath. So why does this all happen anyway? So the CMT, why is CMT such a common problem? And people have thought about this for a long time, and we've basically decided that the reason that CMT, and the reason that even non-hereditary neuropathies are so common, is that these are the longest cells in the body. So the neuron that's going down to my toe sits up about there in my spinal cord. That's about 3 feet long. So that's the longest cell in the body. And so you can imagine that if there was something bad that's going to affect long cells, it's this cell and its connection to your toe that's going to take the worst of the problem. And there are a couple of reasons that we figured out why that might be true. The cell's very long, and so if something's made up here in the spinal cord, it's got to get to the toe. That can take a year. And so if anything messes with that transport, that's going to be bad. Excuse me. Excuse me. We know that the energy that those mitochondria are making have to be made all the way along the way. So if you have an energy failure anywhere along the way of this long axon, that's going to be bad. So, and then finally, we just figured that the cell, not only is it the longest, but because it's the longest, it's the biggest cell in the body. So neurons that are, oh, well, thank you, John.
First blue button on the end.
It's kind of a, that's a Wisconsin pointer, it's red. I'm a Michigan guy, I'm going blue here. I'm in Texas. I should be orange to be a longhorn, right? Is that? What's that? Red? Red in Texas? Susan, now you're pointing to a glass. You gotta. Oh, thank you. I knew we'd get those college jokes in here sooner or later. So we think that this is the fundamental problem that makes CMT a problem for axons. Thank you. You can imagine how much fun it is to sit with me all day on Saturday on a meeting that Pat leads. And now I want to talk a little bit about the CMT1 cause. So this is that special electron microscope picture of the axon. Those are the neurofilaments. These are the mitochondria again. And this special wrap, that's a set of layers of specialized cell membrane. And so things that mess up this specialized cell membrane cause a myelin problem. And that turns out to be probably the most common cause of CMT going. You have too much of a protein called PMP22 in this myelin, and this whole myelin sheath becomes unstable. You have mutations that affect a protein called myelin protein 0, which also resides in this myelin sheath, and the myelin sheath becomes unstable. So we think that problems in the myelin are the cause of the CMT1s. And now finally, we're going to turn to the 3 kinds of CMT I've been charged with. So CMTX turns out to be the second most common form of CMT. Maybe somebody in this room even has it. It's caused by mutations in a gene called connexin 32. And the way that we've thought about approaching this, and I'm going to show you the reasons why in a second, is that we think that the loss of connexin 32 in the myelin sheath causes CMTX. And so if it's the loss of this gene working in the myelin sheath, then we think that maybe we can replace the gene in the myelin sheath and have that be a treatment. And though I'm not going to show you any data, there's a colleague of ours who we're going to recruit this year, I'm telling you, who's got a gene replacement strategy that's worked amazingly well in the mouse model of CMTX. Rudolf Martini is a scientist in Germany who's shown for the last 10 years that somehow inflammation not the actual gene defect, but inflammation that causes to happen, accelerates or worsens CMTX. And he's got an amazing story of how blocking that inflammation actually improves CMTX, and the CMTA has funded Rudolph's work on this approach. So I told you we have the animal model, and Mike Scherer has led, for doing a clinical trial, an important piece of work showing how CMTX affects people and how it gets worse over time. And you sort of need to know that. If you're going to treat somebody for a kind of CMT, you sort of need to know for their kind of CMT how they get worse over time. And for CMTX, we actually know that. So this shows fundamentally the problem. Here's the normal nerve that I showed you once before. Here's the nerve of that patient with the skinny legs I showed you. So something's gone on here. He doesn't have all these nice, big, healthy, happy-looking axons. He's lost a lot of them, and the ones that he has aren't working so well. So fundamentally, the problem with CMTX is that the nerve should look like this, becomes affected like that. And why is that? It's because this protein here, called connexin 32, isn't present in the myelin sheath. Here's where it should be. These pictures these are nerve fibers that have been stained to show you where the connexin 32 is, and you can see it's in these special structures right here. They're called incisors. If you don't have these connexin molecules localized in your myelin, then they form a special thing called a channel that allows things to pass through it. And without those channels in the myelin sheath doesn't work right, and basically what should happen normally is that we're going to say proteins and sugars should go right through these channels and feed the axon. And if you don't have connexin 32, this doesn't happen. So we think that connexin 32 forms a functional pathway in the myelin sheath. If you don't have it, the axon starves to death. You get this axonal disease, and you want to fix that. And if you could replace the connexin 32 in the myelin sheath, you would fix that. So that's exactly what we did, just to show you we can cure a mouse. We made connexin 32 be expressed in a mouse. I'm going to show you on the right, this looks like a healthy, happy nerve. Here's what it looks like, a little bit higher power. Here's a littermate of this mouse, and it doesn't look so good. See all these little thin axons here? They just don't look as normal as that. So basically, this mouse got the human connexin 32 gene put into it, and this mouse, his brother, didn't. And the result was I fixed this guy, and he's all better, and this one isn't. So that proves that you can fix connexin 32 in a mouse. And if you could do similarly a gene replacement strategy, gene therapy as it would be called in the popular press, you would fix connexin 32 in a person. And so that's a very incredibly important concept. We just need to figure out how to do it. And if you got Pat driving you, I'm pretty sure we're going to cross that finish line. So second kind of CMT that we're going to talk about is CMT2E. Now this is like alphabet soup, but this is a problem where there's a mutation in a gene called neurofilament. That's the gene that makes those little dots in the picture I showed you. And we know something about the genetics of that. And one of the things that we typically see in patients who have this CMT2E is the neurofilaments make these big clumps. And that's not normal. And so we think that that might be the problem. And so if we can prevent the clumps from forming, then maybe that would fix the CMT. So that's our hunch, and we've worked on that to some degree now. We have acquired the animal model of CMT2E that Ron Lee made, which has a mutation in the gene that causes the human disease. And we are in desperate need of patients with CMT2E to get involved and become part of the natural history. We need to sort of know how mutations affect people at different points of their lives. So if we had a treatment, we would have some way of knowing that the treatment worked. This is just to show you that there are different kinds of mutations of CMT2E that have been described. So it's not just one mutation. And this is to give you an idea that these guys get together and they're friends. So there's a light, a medium, and a heavy, and they form these little friendly clusters. And when all is well in the world, you can actually make a cell. This is Pat's idea where you have cells growing in a dish. If I put the normal light gene into a cell, it makes these beautiful sort of long filaments. And if I put one of these mutations in them, they just form clumps. So the mutation does this, the normal does that. We'd like to make this look like that, and we think things would be better for the patient. This is just to show you that a nerve from a person with CMT2E has these large clumps in it. You can see these weird large structures. And this is from Ron Leem's brand new publication of December 2014, and you can see these green dots that are filling this cell over here. If you look at the normal mouse, it doesn't have those, so we can see that the mouse has the green clumps. So now we're in the wonderful position of, if somebody had a great idea of how to prevent the aggregates of neurofilament from happening, we could put those drugs in a mouse. Those mice are, there's a big colony of these mice at a company that Pat's paying a lot of money to have, so we want some useful use of the mice. So we're going to give those mice compounds that have a chance of de-aggravating these filaments and see if the neuropathy gets better. So that's a totally doable thing, and there are in fact already lots of compounds that people have described that de-aggregate proteins, and so putting those compounds into this mouse I think is the first order of business. And then we're going to talk about CMT4. So this is a little bit more tricky. So CMT4 is a category, which means your CMT is recessive. You didn't get it from your mom directly, you didn't get it from your dad directly, but your mom and your dad had individually one mutation in one of their two genes, and that mutation got together with one in your other parent, and now you have a recessive CMT. So there are probably 20 different kinds of CMT4. So I can't talk about them all in a meaningful way here, but just to say we know fundamentally the problem with CMT4 is that something isn't there that should be there. And in most of the CMT4s, it's something that needs to be in the Schwann cell. So if you could take the approach that I described for CMTX, that is, all right, you're missing a gene, I got this gene therapy trick, I'm going to put the gene back into the Schwann cell and make well out of the world. I think that would be a completely reasonable approach. So there are genes that have been identified, and I'm just showing 3 good examples here. There's a gene called GDAP1. There are these 3 genes that sort of form a cluster of proteins. And there's a gene, I won't even try to pronounce it, it's SH3TC2 as an abbreviation. That's probably the most common form. So at each one of these cases, we have a pretty good idea of what these proteins are supposed to do, but in a sense that may not matter if we don't know what they do, but replacing the gene with a virus does the job, then we can sort of not get stuck on that point. We might be able to rescue the mouse and the person from having the effects of not having the gene simply by replacing it. We have mouse models for each and every one of these genes, so that we have a mouse that doesn't have GDAP1, SH3TC2, MTMR2. We have mice like that. So we already have the means to know if we had an effective treatment that we could put it into the mouse and see an effect. What we really need for CMT4, which is the participation of the world of people out in the community who have it, is we need to sort of examine them in our clinics. We sort of need to have a numerical representation of how their neuropathies affected them, so that if we have a treatment, we have a means for judging whether the treatment works. So this is where we come back in that circle and we say, okay, we need the patients to be part of our answer here. So the last few slides are sort of intended to drive home how you might do that, besides, you know, coming to these meetings. There's a website, if you look in the corner here, if you Google that, Inherited Neuropathies Consortium, you will find it. There's no other hit on the website that's going to come up like this. And this is the page that you're going to see. So anybody in the world, anywhere in the world, if they can read English, can see this website and join a study that we're talking about. So there is a study that's involved with just finding patients who have different kinds of CMT and basically asking them a series of questions about what kind of CMT they have and how it's affected them.
Can you tell us that website again?
Inherited Neuropathy Consortium. There. And I should point out, this is, I'm very involved in this, and you've already heard Mike Schei's name about 10 times already, and Mike Schei is the leader of this effort. It involves people like us everywhere in the world. And if you believe in governmental support, all supported by the federal government. They are supporting orphan diseases, and this is one of their big efforts. So I'm just arrowing right on that website. If you click on that blue link, you can join a study and self-report that you have CMT. And so if we had a treatment, for instance, and we were trying to reach out to people anywhere in the world, let's say you have CMT4C, to pull one out of a hat. You've self-identified in this website that you have it. We can send an email totally, legitimately to you and say, do you want to be involved in a study? If we had a treatment, this is how we're going to try to recruit patients. This is just the page that you would see if it came up. I'm not going to be able to take you through all of it, but basically there are different studies that you can be involved with already, and in the magic day when we have a treatment, we'll send emails out through this as well. And if you find anything that I said to be confusing, just try to read this and you'll really be confused because I wrote it. This is a website that we have written for physicians and scientists and researchers, where I've tried to explain in a page for each kind of CMT what's known about it so that somebody could have access to it. I've recently started my rewrites, and so I'm going to bring this up to date over the last 4 years. And basically we're just trying to tell every expert in the world that wants to know, this is a one-stop shopping, what's the skinny on CMT1A, 1B, etc. More palatable are the things taught over here, because that was written by a genetics counselor, and they speak in better terms than do I. They simplify things down, and you can learn about hereditary neuropathies written by two genetics counselors who worked with Mike at Wayne State. So I really encourage you to visit that website, because that's sort of a portal to the future. I mean, it's got some useful information on it for where things are, but it's going to be the way that the CMTA is going to work to reach out to patients when we have medications for different kinds of CMT that we're hoping to find. So I probably ran too fast. I have a few minutes left over. I could take some questions, or we could, where are we at with time? I lost Susan.
You have about 10 minutes.
Okay. Would that be all right to take a few questions?
I
Please.
My daughter, she has CMT, and nobody in our family has it, on my side or his side. And the doctors can't, they told us it's CMT1, and then when we told nobody in the family has it, then, okay, it could be CMT2. They don't know in Amarillo, they don't know anything about her disease.
So if you work with Susan, we'll get you the name and an appointment at a CMT Center of Excellence. So there are not enough CMT experts in the world. I've trained 3 in my career, and so it's a one at a time kind of a deal. There aren't enough people, and there certainly isn't anybody in Amarillo, Texas that I could give you the name for. So you might have to do travel, but if you're willing to travel, you can definitely get with a CMT Center of Excellence, and you can definitely get a diagnosis. At this point, everybody with CMT should be able to get a diagnosis, even if it involves doing stuff that's at the research end. Like we have the ability to sequence all 20,000 genes in a person and deduce which gene it's caused. And particularly if you have a child and you and your husband aren't affected, you might have recessive, and you'd have to sequence maybe all 3 persons' DNA, but you could even find a new cause.
They did like a testing, like a nerve testing
Yeah.
on me and her father, and they, nothing. They
So
didn't have,
we probably don't have in 10 minutes the time to do a full consultation here, but
right,
I'd certainly happily see you. And I've got 10 colleagues who would do it too.
okay, I appreciate that.
Yes.
Thank you.
Are these different websites in our literature that we have received?
I don't know that. Maybe not, but it wouldn't be hard to find. If you're here, you can follow up with a phone call and an email, and we'll get it to you.
I have CMT1X, runs in my dad's family, at least 4 more generations back, and our daughter has it too. I'm just wondering if you have any idea how long it will be before you have the human model or tool that you talked about. You said you can do it in animals now, in a mouse, I guess.
So the simple answer is that if we're doing gene replacement as the way to go here, doing that in the mouse has just been done. It's not even published yet, so you're hot off the press. It's not even, so
Okay.
the means to do that, if that really pans out, would be years to get it to work. You'd have to do, there's a number of steps in between, but it's the fact that it can be done. Take heart in that.
Good morning, thank you. I'd like to do a little networking. I allegedly, as of the last 90 days, have been diagnosed with 2D. And
2D.
GARS.
GARS.
And I wonder if anybody in the room is in that boat.
Probably not, if I had to make an educated guess. It's a pretty rare form, but if it's what you got, it doesn't seem very rare, does it?
Correct. And it's new, late onset. Any comments on what you guys are looking at? There's so many variants. I read 80, and obviously we're the.
Well, let's talk at lunch. There's no simple answer to what you just asked me.
Hi, I just wanted to say that the MBA has a clinic down at UT Southwestern, and they're pretty good at dealing with my CMT. So they're out there for you guys. Thank you.
It's good to know.
Thank you, Dr. Scherer. Would you suggest that somebody get genetic testing first, or would you suggest they go to the center, one of the centers first, and then get genetic testing there? Because right now, I mean, I don't know about everybody else, but in the Dallas-Fort Worth area, if you go to the MDA clinics, they basically just tell you it doesn't help to have the genetic testing. That's what they've told me at both clinics. So I'm just wondering what you would suggest.
Well, it's sort of a philosophical point what they might be telling you, you know. So until we have a treatment, people could always take that line. But on the other hand, I would say, unless we know what kinds of CMT there are and what kind you've got, how the heck are you going to benefit? So we're going to wait until we have a treatment for CMT before everybody gets tested? That doesn't sound, to me, very compelling of an argument. So, you know, until recently, the economic cost of doing genetic testing was the impediment. But I want to give a shout out to GeneDx. They've changed the game. They have changed the game. So maybe we'll stop here and.